So there are two different types of myofilaments that we're going to focus on, and these are both microfilaments. The first one is called myosin, and this is also the thick filament. So these are two different names. They're interchangeable. Something is either it's thick filament, that means it's myosin. So here's myosin up here in the top. You can see one single myosin filament here, but myosin looks more like this in the actual muscle. And it's got these little heads that I think look like golf ball clubs. And these are actually going to grab onto the actin and pull it in one direction to cause contractions. The other type of microfilament are the thin filaments because they are physically smaller. So this is a much closer image right here of actin. So this is the thin filament, and you can see that it's only 2 strands. And covering the actin are these proteins called troponin and tropomyosin. So tropomyosin is a long cord on the actin, and then there are little troponins all along. These look like those little dog food treat feeders. So tropomyosin and troponin are regulatory proteins. When they're in one structure, so the way they are right here, they're covering the active site, you can't have contraction. In order for a muscle to contract, those are going to actually move out of the way, and then the myosin is going to grab onto the actin with its little golf ball clubs and pull on the actin, and that's how it's going to cause a muscle contraction. Keep in mind that when muscles contract, so here's actin and here's a myosin, these are just going to move past each other to make the entire thing shorter. The actual myosin and actin microfilaments don't change shape, they just slide past each other. It's called the sliding filament model. There are a couple of other filaments or proteins in here that are important. So there's an elastic filament called titin, and this was discovered fairly recently. And you can see titin is this kind of green curly protein here. It actually connects the entire sarcomere together from one end all the way through the middle of the myosin, all the way to the other end of the sarcomere over here. The reason it's called titin is because it is the largest protein that we know of right now. And then there's another one over here called dystrophin, and this is an anchor. So you can see here, this is my sarcomere down here with my actin and my myosin and my titin. And this is my plasma membrane, right, that sarcolemma. And the dystrophin here kind of creates a connection. So this is going to mean when this contracts, it's actually going to pull this with it. So that the entire muscle contracts from the inside all the way to the outside. So here's a sarcomere. And the sarcomere is what causes the banding pattern striations. So you see striations in skeletal as well as cardiac muscle because they have organized sarcomeres. This is the functional contractile unit, and these are in sequence, which means there's one sarcomere here, there's another one here, there's another one here. Each one of these is going to shorten. So this one's going to shorten this way, this one, and that's what causes the entire muscle to shorten, because each individual sarcomere is shortened. So there are some structures on here that you should know. There is a Z disc at the end of each sarcomere, and it's called Z disc because it looks kind of diagonal. When you look at it as like a 3D structure, it looks a little like a waffle fry. So there's one there, and there's a Z disc here. So that determines the end of the sarcomere. So a sarcomere runs from Z disc to Z disc, and everything in between is one sarcomere. So you can see there's one sarcomere here, there's one sarcomere here, there's one sarcomere here. Inside here, we have myosin and actin. So our thick myosin is right here, and it runs right through this M line. So M for middle. All the way over here. Notice the myosin doesn't run all the way to the Z disc. It's connected to the Z disc with that titin elastic filament. And then the actin starts on the Z disc, but it doesn't run all the way to the middle. So there's space between the actin filaments. Those are actin. When you organize it like this, it gives kind of a banding pattern. So there is a dark band called the A band. So I remember this because dark has an A in it. So here you can see the A band, and on a microscope that looks darker. So you can see 3 dark bands here. This is caused by this overlap in the filaments where we have actin, myosin, actin, myosin, actin, myosin. So because there's a lot of filaments from here to here, it creates a dark look. The light band is called an I band, which I remember because light has an I in it. So here's my light band, here's my light I band. And that's going to run from myosin to myosin, so it actually runs over the Z disc. And that's because in this region here, there's no myosin, it's just thin filament and titin, and that's not as dense, there's not as much stuff here. So these areas tend to look light under the microscope. The H band is inside of the A band, so you can see it right here. It actually looks kind of like an H in the center here. And this is where there's myosin, so it still looks pretty dark, but there's no actin, so it's a teeny bit lighter than the surrounding area. When the sarcomere shrinks, so when it contracts, the actin and the myosin are going to overlap each other more, and the I band and the H band are going to get smaller, and the A band is always going to stay the same size because the myosin does not change size. So again, actin and myosin, they don't change size. What they do is they just slide past each other. And there's a maximum amount of distance this can contract, right? The actin can't really slide any further than that midline, and the Z discs can't come further than the myosin. So this has an amount of distance it can shorten. If you stretch this muscle too far, the actin and the myosin won't be close enough to each other to pull against each other. So the way it contracts is the myosin actually grabs the actin and pulls it in towards the center. And if you stretch the muscle too far, then it wouldn't be able to do that. So take a moment and describe the structure of a sarcomere as much detail as you can. A picture is worth 1,000